Thanks, Ren, for the introduction. I guess the first thing I should say is one of the very few non-US companies here today is just to flag Gil's sterling efforts with the Alliance for Regenerative Medicine, in particular the establishment of a European chapter for that organisation. As I'm sure Gil would agree, it's important that this field talks together globally across boundaries with one voice, and that was a key motivation in Regeneron becoming a founder member of the European chapter forearm. So I'm delighted to have done that alongside Eduardo Brava at Tygenics, and there are a number of European companies now signing up to that European chapter. And of course, the aim here is that we do cross-fertilise ideas at a regulatory level, at a technical level, and so on, that affect us all. It's clear from my perspective that the US, certainly at a commercial or applied level, very much dominates the field at the moment, as is apparent here today. But nonetheless, the European side of things is difficult, intractable, but a major market, and one characterised by very differing views across national boundaries. So it's important that ARM, I think, gets at that, and as I said, we're delighted to be a part of that. We're a public company, so there's the usual disclaimer, which I'll test you on at the end. So we're a public company quoted in London. Our market cap tends to move around, but it's averaging around 50 million US at the moment, a little higher at the moment, actually. But that's where we've been. So we're a very small business in terms of market cap. The ownership is kind of broken down as follows. Quite good fund and institutional shareholdings now, so we're reasonably confident at raising money when we need to. We don't do large raises, but we are pretty successful at raising what we need to move us forward to the next milestones, and that we've been doing for around 6 years now on AIM. We have three core assets, two of which are therapeutic, and only one of which I'll cover today, which is the CTX human neural stem cell line. That's in clinical development now for stroke, REM1, which I'll talk about, and in preclinical development for CLI and a number of other indications, and I'll touch on CLI very briefly at the end. We do have a human retinal precursor cell line as well, which is in preclinical development for a range of retinal diseases, initially focused on RP, but I won't be covering that today given the nature of this particular panel. And finally, a range of non-therapeutic neural stem cell lines called RenCell, which we licensed to Merck Millipore for sale globally a number of years ago. We are an allogeneic business model player, and we use proprietary cell expansion selection technology that we've developed over the last 5 to 6 years plus, which means we can easily scale and manufacture our products to serve the large patient populations we're targeting at what we believe will be an attractive cost of goods, and I'll come back to that specifically. And in common with other companies, we do make wide and, in fact, increasing use of collaborative relationships with a number of academic institutions, both here in the US and back at home. So CTX is a conditionally immortal human neural progenitor cell line, fetal-derived, and we've applied our patented expansion technology to it, and that technology is chemically controlled, so it's a modified cell approach, but it gives us unlimited supply from a single tissue sample. And over the last 5 years or so, we've laid down multiple cGMP banks across a range of contract manufacturers, both in the UK and here in the US. So we know that third parties can work with this technology straightforwardly and grow cell banks. And that provides the seed stock for all the clinical and commercial product that we need going forward. And the cell characteristics are fully maintained throughout that cell expansion process. As you might imagine, the regulatory authorities have looked at that exhaustively, and therefore we've tested exhaustively this technology at a number of levels over increasing passages, sufficient to give us the cell banks that we need. Cells are non-immunogenic. The thawed product, the fresh product, has a 96-hour shelf life. The frozen product, obviously, much longer. And we've demonstrated consistent efficacy preclinically in stroke and ischemic limb models over the last sort of 5 years, 6 years since we started testing CTX preclinically, initially in stroke and more recently in CLI. And we know that we believe it has potential in other neurological and vascular indications as well, and we're in the process of testing preclinically there as well. It has a clean, acute and chronic tox, tumorigenicity and CARC profile preclinically. Again, we've done a raft of acute and long-term safety studies ahead of taking the program or CTX into our first clinical trial. And that is a clinical trial in stroke in the UK, so it's been through the MHRA GTAC approval process. And For those of you that wish to know more about what the regulatory process is like in the UK, based on one or two of the comments that were made a short while ago, I'd be happy to take that offline. It's an intriguing process one has to go through now, especially dealing with the Gene Therapy Advisory Committee. We have some stories to tell there. And perhaps in common with most of the other companies presenting today, we're also great believers in the multimodal effects of stem cells. And indeed, although this is a neural cell line, a lot of the mechanisms of action that we see are very common to other cell types, including mesenchymal cells, of course. So what's our strategy for taking CTX forward? Well, we're taking it forward initially as a treatment for stroke disability clinically, and also looking at it in an earlier stroke setting, subacute stroke, and other neurological conditions and CLI. And really what we want to do is leverage the mechanism of action of these cells where relevant and use the existing preclinical data package that we've used wherever possible. And that, we hope, will lead to a faster regulatory process now that we're in the clinic to build an emerging pipeline of high-value therapies, and in so doing reduce dependence on the success of any one of them. And we fully recognise the fact that we have a relatively thin pipeline at the moment, 3 or 4 key programs that we're investing in, including the stroke program. But we would like to do more, and funding clearly does become an issue at some point. But nonetheless, that's what we hope to do with CTX longer term. At a clinical level, to fast-track it in relevant patient groups, and I'll come back to that notion in terms of the stroke program in a second. And again, in common with what Silvio and others have mentioned this morning, we do believe in seeking commercial development partners as we gain clinical POC data, and also further collaborative relationships beyond that to really optimize what we're doing in terms of technology. There's a lot that still needs to be done. If we're honest, in terms of developing good potency assays, bioprocessing, actually making improvements to the manufacturing process in the longer term. And we do that collaboratively wherever we can. We're less than 20 people as a business, so most of what we do is outsourced. We do, though, wish to retain control over the expansion technology in the cell manufacture as we move forward commercially. We do believe in keeping control over the manufacture of the cells at a commercial level in terms of rights. IP is important to us as a business. We've worked on this over a number of years. We have an extensive patent portfolio covering composition, method, delivery, and use of our key cell line assets. So the cell lines themselves are patentable entities. We have licenses over the cell expansion technology, or key elements of the expansion technologies, and access exclusively to a number of other neural and retinal stem cell patents as well through license arrangements, both with commercial and academic organizations. So a very broad patent state as far as we're concerned, something we're very comfortable with. And in terms of manufacturing and cell formulation, again, trade secret coverage for a lot of that stuff. So moving on to stroke very quickly. Again, I didn't think I'd be the first one on in stroke, actually. Well, I suppose Gil may have given some indications on this earlier on anyway in his broader talk. It's a very large indication, clearly, and an unmet need. And initially, we're focused on ischemic disabled stroke patients. So if you get a stroke and survive it, you've got a 50-50 chance of being left disabled by it to a greater or lesser extent. And those are the patients we're targeting with REM1 initially. These patients occupy, it's a classic hospital bed blocking indication, and the health and social care costs are obviously enormous, as previously pointed out. No treatments available beyond the acute phase TPA for these patients. And we've always believed that despite the conventional small molecule approach being singularly unsuccessful in stroke or acute stroke, we've always believed that a cellular intervention might yield a result, and certainly preclinically that's what we've seen. Obviously a very large commercial market in this indication. So we have shown and replicated a number of preclinical studies showing that CTX does reduce quite significantly stable state motor deficits in the stroke rat. And just by way of a very brief snapshot, we actually published a study last month, which was done at Mike Modo at King's. Mike's now at University of Pittsburgh Medical Center. And this was a classic MCAO stroke model, primarily focused on motor as opposed to cognitive deficits. And we tested the cells in a number of settings, including where the cells were injected. This was intracerebral injection, putting them in a number of different locations in the brain and seeing if that made a difference to the degree of recovery seen. And it is, for our cells at least, important. The lesion topology is an important driver for the degree of efficacy seen, as is the site of implantation of the cells. So these data, to the extent that they can be predictive of clinical endeavours, does tell us something about how we should be deploying the cells at a clinical level. And this is one of about 3 or 4 studies that we've published looking at MCAO stroke models with CTX. So moving on to the clinical experience, we started a phase 1 trial in Scotland about a year ago. It's a 12-patient, 24-month follow-up dose escalation safety study. We are looking at a range of efficacy evaluation measures as well, as you see in the table below. These are classic stroke measures, but with quite heavy use of structural and functional MRI as well over time. It's straightforward intracerebral injection, so we're putting these cells at relatively low doses directly into the striatum. It's a straightforward neurosurgical procedure, typically involves a 1 or 2 night hospital stay. We're not immunosuppressing the patients, and it's being run out of a single site at the moment in Glasgow under Professor Keith Muir at the Institute of Neurosciences and Psychology at the University of Glasgow. So we're 5 patients in thus far. So far, no acute or longer-term cell-related safety issues. We're hoping to treat the remaining dose cohorts, 4 dose cohorts in this trial, over the course of this year, and moving into a phase 2 multi-centre study next year. And really the strategy is to try and fast-track clinical development in targeted patients. We're somewhat different from other approaches because we are, first of all, putting cells in directly, and we do believe that at lower doses, the cells are going to engender an effect more specifically in certain types of stroke. And I'm talking really about subcortical deep brain strokes, lacuna strokes. And you can actually see this in the third of the 5 panels. These are patients 1 to 5 in order. And you can see pretty severe damage, cortical and subcortical, in the first patient, but very minor deep brain damage in patient 3, but both manifesting quite severe disability for these 2 patients. And certainly anecdotally at least, we're seeing what appears to be a level or a greater level of biological activity in the third patient, to the extent that we can actually attribute anything to what we're seeing so far. Obviously, this isn't an efficacy study. In terms of what we are seeing, just a couple of snapshots presented by the PI at the Stroke Forum last month in Glasgow. First of all, an NIH SS, 5 patients in, and obviously some sort of modest improvements on stroke scales, but these are very low doses, 2 million cells, 5 million cells in the second dose cohort. So sort of trending nicely. Obviously, when we get to the higher doses this year, we'd hope to see something more significant perhaps, or something that at least indicates what we might see at higher doses for a phase 2 study. And similarly on Ashworth scales, a measure of spasticity in these chronic disabled patients. Again, certainly no deterioration over time, and some modest improvements both on the formal measures and anecdotally as well in 1 or 2 of the patients, interestingly, at these low doses. So a good start to the trial as far as we're concerned. Recruitment's quite tricky. It's a very stringent, there are very stringent inclusion-exclusion criteria, but nonetheless, we're very confident that REM1 will produce a significant effect if we can target it appropriately at the right types of patients. Very quickly on CLI, again, a very significant unmet need, largely driven by diabetes, one of the most significant contributing factors to PAD, of which CLI, of course, is the end stage. So this is about avoidance of amputation and the costs that go with that. We've developed or generated quite a raft of preclinical efficacy data using CTX, interestingly, beyond the brain with the guys at Bristol Heart Institute. And again, this is playing very much to the pro-angiogenic nature of the CTX cells. We presented data at AHA and Diabetes UK over the last couple of years or so, and we're hoping to start a phase 1/2 clinical trial later on this year. And just a quick snapshot preclinically of what we've done here. And again, this will be familiar to the other CLI guys in terms of what you're looking for preclinically, in terms of endpoints for a preclinical study, in terms of Doppler blood flow, increased capillary density, and so on. Finally, just a quick nod to my colleagues back at base and our clinical advisory board as well. And with that, I will leave it. Thank you.